The bacterium that has invaded human body secretes some molecules called as chemoattractants. These chemoattractants are N-formylated peptides. The human immune system has neutrophils. The surface of neutrophil has some G protein coupled receptors. The chemoattractants released by the bacterium then binds to the G protein coupled receptor present at the front side of the neutrophil. These bound chemoattractants are recognized as foreign by the neutrophil. After the chemoattractant binding, the G protein coupled receptors become active. The active G protein coupled receptor then stimulates a G protein called as GI. The GI is trimeric in structure. The stimulated GI then stimulates the formation of PIP3. The PIP3 then activates RAC protein. As a result, RAC protein dominates. Due to this RAC protein domination, the actin polymerization occurs. This actin polymerization causes a protrusion at the front of neutrophil. The second messengers like PIP3 is rapidly converted to PIP2 in presence of active lipid phosphatase. So the PIP3 is short-lived. Due to this short-lived PIP3, the protrusion is limited to the region of the neutrophil closest to the chemoattractant. The chemoattractant bound active G protein coupled receptor also stimulates G proteins G12 and G13. Here, these two G proteins are denoted as G12 and 13. The G12 and 13 are trimeric in nature. The stimulated G12 and 13 then triggers the activation of Rho protein. As a result, the Rho dominates at the back of neutrophil. Due to this Rho protein domination, actin-myosin contraction occurs at the back of neutrophil. This actin-myosin contraction causes a cellular contraction at the back of neutrophil. Whenever the Rho protein dominates, it blocks PIP3 of the RAC pathway. So we can say that actin-myosin contraction blocks the actin polymerization. Whenever the RAC protein dominates, it blocks Rho protein in Rho pathway. So we can say that the actin polymerization blocks the actin-myosin contraction. Here we can also say that RAC pathway and Rho pathway are antagonistic to each other. The protrusion at the front of neutrophil and the contraction at the back of neutrophil help in directed movement or polarization of neutrophil. Now, important points. The G protein coupled receptors on neutrophil can detect a difference of only 1% in the concentration of N-formylated peptides on one side of the cell versus the other. Please like, subscribe and share.